Hello, and welcome to a Taconic T-Onco webinar entitled Preclinical Evaluation of Anti-Metastatic Drugs, given by Drs. Caroline Mignard and Xavier Tizon from our French oncology partner company, Oncodesign. First, I want to introduce myself as the host of today's webinar. My name is Megan McBride, and I work in Taconic's Global Business Development team, where I am responsible for commercialization of genetically modified models. I will be your moderator for today's webinar. Before we get started, I'd like to address a few commonly asked questions for this type of presentation format. Let me introduce today's speakers. After obtaining a PhD in biology from the University of Strasbourg in France, Caroline Mignard worked as a preclinical project leader in a contract research organization in Strasbourg for 4 years. She then joined Oncodesign in 2004 as a study director in preclinical research, and she is now the director of Oncodesign's Biological and Chemical Resource Center. Caroline continues to supervise experimental studies for Oncodesign's customers, particularly involved in studies based on mice reconstituted with human immune systems or engrafted with patient primary tumors, PDX. Xavier Tizon is a trained biomedical engineer who completed his PhD in medical image processing in 2004. Today, with more than 16 years of experience in medical imaging, he has been the head of the imaging lab at Oncodesign for 7 years, where he routinely uses anatomical and functional imaging and assess the efficacy of anti-cancer therapies. We hope you enjoyed today's webinar. Caroline?
Yes. Good morning, good afternoon, everybody, and thank you, Megan, for this introduction. Despite many years of research aimed at suppressing tumor growth, metastasis remains the primary cause of death from cancer. Patient prognosis is connected with metastatic disease, as reflected in the staging system for many types of cancer. The diagnosis, metastatic cancer, is considered terminal for most cancer types. Mechanistic understanding of the metastatic process and development of anti-metastatic therapies are urgently required to improve survival in cancer patients. The challenge of preclinical evaluation of novel anti-cancer compounds is to develop more relevant models, mimicking the disease evolution in humans, and use non-invasive imaging techniques that allow the monitoring of tumor progression and dissemination. This presentation is divided into different parts. Firstly, I will describe briefly what is metastatic cancer. Then I will describe symptoms of metastatic cancer and therapies. In a third part, I will expose several examples of animal models used as tools to evaluate new therapies. Xavier will continue the presentation with a description of various imaging methods for evaluating new therapies. And finally, some conclusion and perspective will be drawn. Metastatic cancer is a multi-stage process in which malignant cells spread from the tumor of origin to colonize distant organs. Metastatic cancer has the same name and same type of cancer cells as the primary tumors. For example, colon cancer that spread to the liver and form metastasis is metastasis colon cancer, not liver cancer. Most cancer, including cancer of the blood and the lymphatic system, can form metastases. The following table shows the most common site of cancer metastasis, excluding the lymph nodes, for several cancers, breast, lung, colorectal, etc. The most common sites of metastasis are the bone, lungs, and liver. To establish metastasis, tumor cells must undergo a series of events, including local invasion, meaning cancer cells invade adjacent normal tissue, intravasation, escaping an oikis, that is detachment induced cell death and entry into the circulation. Disseminated tumor cells manage to survive in circulation before they can arrest in a distant organ and leave the circulation, that is extravasation. Cancer cells multiply at the distant location to form small tumors known as micrometastases. Through angiogenesis, micrometastases stimulate the growth of new blood vessels to obtain the blood supplies for continued tumor growth. The success of this process depends on the properties of the tumor cells and the propensity of the host organ to accept these cells. This organ tropism theory was first explained in the seed and soil hypothesis by Paget, more than a century ago. The course of metastasis can vary according to the tumor type. Metastatic cancer can stay dormant at the distant site for many years before growing again. For example, that is the case for breast tumors. Cancer cells can be competent to infiltrate distant organs at early stage, but their frequency enter a period of latency before colonization. When symptoms of metastatic cancer occur, the type and frequency of the symptoms depends on the size and location of the metastasis. For example, cancer that spreads to the bone is likely to cause pain and bone fractures. Abdominal swelling or jaundice can indicate the presence of liver metastasis. Treatment for metastatic cancer are currently similar to those used for primary tumors. It can be treated with systemic therapy, such as chemotherapy, targeted therapy, hormonal therapy. Metastatic cancer can also be treated with local therapy, such as surgery or radiation therapy, or a combination of these treatments. The choice of treatments generally depends on the type of primary cancer, the size, location, the number of metastatic tumors, the age and general health of patients, and the treatment the patient received in the past. New therapies are in development. New ways are studied to boost the potency of immune response against tumor or to disrupt individual steps in the metastatic process. The development of targeted therapies, therefore, required the identification of good targets, meaning targets that are known to play a key role in cancer cell dissemination. The number of targets implicated in metastasis process is very huge. It would be boring to describe here all of them. We can list a few examples of therapeutic strategy currently in development. The tumor vasculature can be targeted with multiple drugs, such as bevacizumab, which targets VEGF, or CXCR2. Immune activation, marked by an activation of T cells, can be achieved through a blockade of CTLA4 or PD-1 receptor. Immune cell recruitment and expansion can be blocked through inhibition of cytokine axis, such as CXCR4. Xavier will expose results of imaging CXCR4 expression in orthotopic breast xenografts with this compound, AMD 3100. Cancer metastasis is a complex process. It is unlikely that complexity of the human disease can be entirely modeled by one system alone. It is possible to choose the appropriate model for study of individual questions. Preclinical model can be divided into two different groups, syngeneic models and xenograft models. The main advantage of syngeneic model is that the transplanted tissue, the microenvironment, and the host are for the same species. This is particularly important when considering the close interaction between tumor and host, characterized by the process of metastasis. The major limitation of the xenograft models is that the effect of an intact immune system on the onset of metastasis and progression are not replicated. Secondly, the appropriate selection of a spontaneous or experimental model of metastasis depends on the specific steps of metastasis being studied. Spontaneous models of metastasis involve the inoculation of tumor cells into an orthotopic site or subcutaneously. Orthotopic implantation of tumors enhance the possibility of distant metastatic spread compared with subcutaneous transplantation. Spontaneous model of metastasis closely resembles clinical disease and permits the analysis of both early and late stages of metastasis. Compared with spontaneous models, experimental metastasis models are rapid and more reproducible, as cells are introduced directly into the bloodstream of the host. They are therefore used for testing therapies targeting late stage of metastasis. A large bolus injection of tumor cells into the circulation by IV or intracardiac route does not closely mimic disease progression in the clinic. And a last approach to model metastatic dissemination to sites that take longer to develop, such as brain metastases, is to locally inject the cells, the tumor cells, at a specific site where they grow as metastases. These experimental models mimic very late step of colonization in the metastatic process. Finally, among all these models of metastasis, spontaneous model represents the more relevant animal model. The main animal model of metastasis available at Tokodesign are summarized in the following tables. The first one for syngeneic model and the second one for xenograft models. The three last models are experimental models of metastasis involving the inoculation of tumor cells by IV or intracardiac route. Most of these models will be described in the next section. So firstly, some example of syngeneic models. The first one is a 41 breast carcinoma model. Tumor cells are inoculated into orthotopic site of bulbs mice. The primary tumor reaches a volume of 2,000 cubic millimeters within 1 month. A tumor spreads to lung and liver. Macroscopic metastases are detected at the lung surface. The mean number in untreated mice are 20. At the surface of the tissue, histological analysis reveals the presence of liver metastases. In this section stained with hematoxylin eosin. Moreover, when mice are treated with indomethacin anti-inflammatory drug, the number of lung metastases is reduced. 41 is a TGF-beta producing murine mammary carcinoma. A number of small molecule TGF-beta receptor antagonists have been recently developed and have shown promise in preclinical studies. These compounds target the TGF signaling pathway by binding the ATP binding pocket of receptor. One of these inhibitors is SM16, showing anti-tumor activity in 41 models. These results have been published in Anti-Cancer Research in 2009. When delivered orally, SM16 inhibits the growth of primary and metastatic 41 tumors. A second spontaneous model developed in immunocompetent mice is the RENCA kidney carcinoma model. Tumor cells are injected into the kidney. Tumor-bearing mice died within 1 month. As early as 3 weeks post-inoculation, metastases are detected in at the surface of lung. The survival time is increased in mice treated with sorafenib, known as anti-angiogenic agent, and the number of lung metastases is reduced. The lung weight is also decreased in sorafenib-treated mice and can be used as readout of anti-metastatic efficacy. Gemcitabine is also efficient as anti-metastatic drug. The number of lung metastases is highly reduced. Primary tumor can be evaluated using MRI without contrast agent for morphological analysis. This imaging method permits also the evidence of lung metastasis as early as day 9. In the same manner, the sagittal image shows the primary tumor on day 29 and lung metastases are shown on this axial image. Tumor-associated macrophages, called TAMs, produce numerous factors that directly or indirectly promote tumor growth. Inhibition of tumor growth through elimination of TAM represents a potential therapy. Macrophages are F480 positive cells and can be detected within the tumor using immunochemistry analysis with F480 staining. This analysis shows that RENCA tumor possesses moderate TAM levels. This readout can be used as tool to evaluate therapy targeting TAM in this tumor model. The last example of syngeneic model is LLC1 Lewis lung carcinoma model, an experimental metastasis model. In this case, tumor cells are injected into the caudal vein of mice and spread into lung. 2 weeks after tumor cells inoculation, examination of lung demonstrates the presence of lung metastases. Cyclophosphamide alkylating agents result in a high inhibition of tumor dissemination into lung. Secondly, we will expose several examples of xenograft model. The first example is autotopic HCT 116 colon carcinoma model. Mice were autotopically injected with tumor cells at D0, and in this example treated with CPT11 from day 13. Survival time was increased in CPT11 treated mice. If mice are terminated 1 month after tumor cell inoculation for waiting primary tumor and counting liver metastases, results demonstrate that CPT11 reduced in a dose-dependent manner the weight of primary tumor and the number of liver metastases. CFRA21 is a fragment of cytokeratin 19 and serves as a biomarker in several epithelial malignancies. It was quantified in mouse plasma over the time. CFRA21 level is increased over the time in control mice. This increase is largely reduced in CPT11 treated mice. This biomarker serves as a valuable tool for monitoring treatment response. Here, a representative picture obtained by MRI with morphological analysis. At the bottom, sagittal images, we can show the primary tumor, and here see kidney, for example. As the top axial images along the plane represented here by the yellow line, we see primary tumor at day 15, and the metastases corresponding to bright zone are detected on day 42. CML5 is a variant of the human LB1319 melanoma cell line that is generated through 2 rounds of in vivo selection. The human melanoma cell line LB13111 mel was IV injected into nude mice. Brain metastasis that emerged was isolated, dissociated, and reimplanted into new hosts for an additional round of selection. This type of in vivo enrichment procedure has permitted to obtain a highly metastatic CML5 variant. CML5 is a melanoma expressing alpha V beta 3 integrin and CCR7 that can be used as potential targets for development of targeted therapy. The particularity of this tumor cell line is it can be used as both spontaneous and experimental model of metastasis. Indeed, when tumor cells are inoculated into the orthotopic site in the mouse, tumor spread to portal lymph nodes only, and surprisingly, this tumor dissemination is not observed in the rat by orthotopic injection. In the orthotopic mouse model, BCNU and temozolomide alkylating agent significantly reduce the primary tumor as well as lymph node colonization. However, when injected by IV route in the caudal vein in both species, nude mouse and nude rat, tumor spreads to lymph node, bone, and brain. This picture represents kinetic study of CML5 dissemination in the nude rat using imaging of tumor glucose metabolism with FDG micropet. This method permits to detect, in this case, ovary, bone, and lymph node colonization over the time. PC3 human tumor cell line has limited metastatic potential in rats from orthotopic site, but metastasizes spontaneously to bone via the intracardiac route. In 2010, at the ACR meeting, we published results demonstrating effects of Taxol on tumor dissemination. The survival time is increased in Taxol-treated rats. In the right picture, a picture of osteolytic bone metastasis in femur of nude rat after intracardiac injection of PC3 tumor cell line. Biochemical markers of bone turnover, which reflect both formation and resorption of bone, are quantified in serum from tumor-bearing rats. Bone metastases in untreated tumor-bearing rats are associated with a decrease in bone formation markers, and they have elevated bone resorption markers such as TRAPS5B. In Taxol-treated rats, the decrease in bone formation marker is lower in comparison with untreated rats, and the level of bone resorption marker is not increased. As in patients, this biomarker can be used to assess response to therapy. Here are representative picture obtained with morphological analysis by MRI. In femur, bright zones correspond to bone metastases, and imaging of the lung shows absence of metastases in this distant organ. The last example of experimental model is a human BT474 breast tumor cells directly injected into the brain where they grow as metastases. Growth of breast tumor in brain is evaluated by MRI with morphological analysis. Tumor volume can be evaluated over the time. In this experimental model of brain metastasis, control die within 3-4 weeks, whereas survival time is increased in Herceptin-treated rats and some of them are alive at the end of the study. I'm now handing over to Xavier for a specific section about imaging modalities.
Okay, thank you, Caroline. In my part of the talk, I will first just name the few imaging techniques that are available for small animal studies. And then I will describe 3 applications around 3 imaging modalities. First, I will talk about bioluminescence imaging to evaluate tumor burden. Then an example using MRI to image the biology of the tumor and the part of the biology that is relevant to the metastatic process. And then I will present an example of molecular imaging with PET. I will briefly try to give you a hands-on approach to choose the imaging modality when choosing a localization for the metastasis and conclude. So for those into the hard science, this is a list of the imaging modalities that are commonly used on small animals. And this is I noticed that there was a kind of a wave-particle duality on this slide. Actually, on the left, you have a wave-based modality, optical imaging, MRI, ultrasound, and on the right, the particle-based modalities, PET, SPECT, and CT imaging. All of them are characterized by a physical principle, meaning what actually is measured, a temporal and spatial resolution, the possibility to use compounds that change the measured signal, and the list of possible applications with their associated complexity and state of validation. So the purpose of this part of the talk today is not to describe all modalities or application, but to illustrate by 3 examples the utility of imaging in projects where metastasis or the biology underlying the metastatic process need to be evaluated. So first, bioluminescence imaging. Bioluminescence imaging is the process of visible light emission by living organisms as a result of a natural chemical reaction. Luciferin is the substrate for an enzyme, Firefly luciferase, which also requires ATP, Mg2+, and O2. On reaction with luciferin, luciferase produces oxyluciferin, adenosine monophosphate, CO2, and light, forming the basis of bioluminescence imaging. When using bioluminescence imaging in vivo experiments, we first have to create a mouse model by injecting tumor cells that have been transfected to express luciferase. This expression can be conditioned to the expression of another gene, making BLI a good tool to measure gene expression itself. But we will focus today on the simple transfections where luciferase is unconditionally expressed. When the tumor is growing or disseminating in the body of the mice, we image them under an anesthesia after the injection of luciferin. There are 2 things that you have to keep in mind when looking at BLI images, and they are both very obvious when looking at the chemical reaction by which the BLI signal is formed. First, the reaction needs oxygen, so hypoxic tissue in which oxygen is not readily available, like tumors, will show heterogeneity in BLI signal. Second, it needs ATP. It will then also depend on the metabolism of your mouse. This introduces variability between animals and can also make your signal depend on several other factors, like, for example, core temperature or time of day. So these are the kind of data that you get with bioluminescence imaging. So this is one mouse in which we have injected ovarian cancer cells, IP, and it disseminates through the abdomen, and you can monitor the dissemination of the tumor cells over time with the full dissemination after about 4 to 5 weeks. So just to summarize the characteristics of BLI, what is measured is the photons emitted by an intracellular chemical reaction. Spatial resolution actually is a little bit difficult to quantify. It depends on the depth, on the light intensity, meaning on the type of cells you use, and the location you're looking at. The temporal resolution is quite good. You can have an image ready in seconds or minutes. You do use luciferin as a contrast enhancement tool, and as limitations, you need to use genetically modified animals, and the light does not penetrate tissue very far. So the consequence of this is that you get semi-quantitative data, but tomographical approaches are available today that improve a little bit and try to get you better data from bioluminescence imaging. As an example, this is an article from Lee et al. From 2007, in which they have injected intracardiacally PC3 prostate cancer cells with a high incidence of bone involvement, and they have studied the efficacy of docetaxel in this model, and you see that it's very obvious you can measure efficacy with this kind of approach. As a side note, they also have done in this study, I won't show the results, diffusion-weighted MRI to study leg disseminations. I'll talk a little bit about this modality later. Second, magnetic resonance imaging. Magnetic resonance imaging, or MRI, is a technique that uses the properties of water molecules to absorb energy in a very efficient way when subject to a strong, constant magnetic field, a physical phenomenon called magnetic resonance. To acquire an MR image, you place your subject in the magnetic field, which ranges from 1.5 to 3 Teslas for humans, and typically 4 to 9 Tesla for small animals, and up to 11 and even higher fields and for specialized applications. You then apply a series of carefully designed electromagnetic pulses called a sequence, and at the same time you record the electromagnetic state of water molecules as it is modified by these pulses. The pulses create contrast between different types of tissues in the body and allow to locate the signal in 3 dimensions as it is emitted by all individual water molecules. This recording, which actually is a time-varying electrical current, is digitized and stored on a computer to create a 2D or 3D image with the help of a mathematical tool called the Fourier transform. Typical spatial resolution is around 100 to 500 micrometers, with a temporal resolution from a few seconds to several tens of minutes, depending on the application. Contrast agents like gadolinium chelates or iron oxide nanoparticles can be used to broaden the spectrum of applications, but even without them, MRI is probably the most versatile of all medical imaging techniques. It is possible, often without the use of any contrast agent, to make the signal dependent on a number of different biophysical phenomena like magnetic properties of water molecules, diffusion, or perfusion. One of the drawbacks of MRI that is often put forward is its lack of sensitivity, usually considered to be in the millimolar to micromolar range. This is not untrue, but only holds in the context of a direct comparison with nuclear imaging techniques and tracer-based contrast. So this is the machine we have here at OncoDesign and the kind of data you get. And those are examples. Caroline showed you those images already of what can be done in terms of showing metastasis in the lung, in the liver, and in the brain. So a short summary is that you see with MRI, you see the interaction of protons with their magnetic environment. You get a spatial resolution about 0.5 millimeter, and it can take from seconds to hours to take one image. You have several contrast enhancement mechanisms, and a lot of other techniques. Technically, it's challenging. It's typically more challenging than bioluminescence imaging. You get to work in a magnetic environment. and it's, as I said, it's a low sensitivity code. Back to the biology of the metastatic process, we know now that angiogenesis play a critical role in the formation of the primary tumor, but also at the end of the process in the formation of a macro-metastasis. There is an MRI technique that has been in constantly growing use in preclinical and clinical studies to measure tumor vascularity. It's called dynamic contrast-enhanced imaging. So I will briefly describe how this works. Use of a contrast agent is common in oncology studies in order to better identify tumors by imaging the contrast agent leaking through their damaged vasculature. But the pattern of enhancement alone is not specific of a tissue or a disease state. We need to study the dynamics of contrast enhancement, for example, using dynamic contrast enhanced MRI. It is used in patients to give information about tumor aggressiveness, but also as an early pharmacodynamic biomarker for anti-angiogenic and antivascular drugs in clinical trial. This example shows images from the protocol we have put in place at OncoDesign on using subcutaneous tumors. This is a human breast cancer model implanted subcutaneously in the flank of nude rats. We perform continuous dynamic imaging using repetitive 4-second scans. After 30 seconds, we inject the contrast agent through a catheter placed in the tail vein of the animal. The signal we measure can be readily transformed into a measure of the concentration of the contrast agent, which allows to use pharmacokinetic modeling to derive parameters for all voxels in the tumor. The main advantages of the technique are that it is directly translatable to humans. It gives early information on the biological effect independently of changes in tumor size, and it can easily be repeated as it is only minimally invasive. Just a few words about one experiment we did some months ago using DCMRI. So this study was done in an intracranial glioma model called the U87MG, and we studied the use of DCMRI as a pharmacodynamic biomarker of treatment with sorafenib, an anti-angiogenic compound. So what we did is that we pre-selected from a set of 20 rats, we pre-selected 16 rats based on baseline measurements that we did, and we measured the DCMRI parameters and discarded 4 of them based on the value of this parameter. The meaning of this is that we try to randomize and build groups that are as equivalent as possible in terms of the PD biomarker that you will use in the study. So from these 16 rats, you get 4 groups. We will focus only on the 2 groups here for vehicle versus sorafenib, because in this study we also try to understand the difference we could see by using 2 different contrast agents, but we won't talk about it today. You treat with sorafenib 2 weeks from D0 to D13, and then you perform DCMRI at baseline, just the second day of imaging, the 4th day, and after 1 week. And you get this kind of results. So on the top line, you have morphological images from D-1 to D7 in the sorafenib group, and the line in the middle shows the parameter that we measure, displayed in color here. The parameter is called K-trans, and the bottom line shows the time curve of contrast agent enhancement in the tumor. So this is the average time curve in the tumor. So after a couple of treatments, there is no big changes, and then after 1 week of treatment, you see very obviously the changes from baseline to D7. And you can see on the average curve of the K-trans parameter that the variation is very readily visible already 1 week into treatment. So it's an early biomarker of efficacy in that case, and it's a parameter that shows you the permeability of the tumor in close relationships with how aggressive it can be and its metastatic potential. The third and last example I will show is using positron emission tomography. So positron emission tomography is a nuclear imaging technique in which you, sorry about the mess for this one. You have basically a tracer which consists of 3 parts: a targeting molecule, an isotope, and a linker. And the 3 parts represent the tracer. You inject the tracer to your animal system or in patient. The tracer meets its target via its targeting moiety, and you can see where it is in the body because the isotope emits photons that you can measure. So it actually emits 2 photons which are exactly emitted at the same energy at the same time and in opposite directions. For this application, you have several isotopes available. You have carbon-11 with a half-life of 20 minutes, so it's a bit short to organize your imaging study. You have gallium-68, fluorine-18, and you have a range of half-life that range from 20 minutes to 78 hours, meaning that you are able with this technique to label either small molecules with these isotopes or molecules that have a longer biological half-life, for example, antibodies or proteins, using the longer half-life isotopes. The typical application of PET is a tracer called fluorodeoxyglucose, which is an analog of sugar, and you label it with the fluorine atom. And the nice thing about this is actually it's taken up by the cell as glucose is, but it's retained in the cell. So it doesn't go through the Krebs cycle and the glycolysis, and it just gets trapped in the cell. So if your cell does need more glucose, then you can see it accumulating in the cell. So this is an example of a mouse study in which you very clearly see in this coronal image the uptake in the abdomen and in the subcutaneous tumor, and you also see the bladder here in which the tracer accumulates over time. And the most famous example of the use of FDG is it's been used quite some time, but it's even much older. But it's the experiment in which we could see very clearly the efficacy of imatinib therapy in a GIST patient. So what you measure with PET is gamma photons that are emitted at the decay of the tracer isotope. You get a spatial resolution of several millimeters. It takes minutes, tens of minutes to have an image. You have available short and long-lived isotopes, and in terms of limitation, you have quite low spatial resolution. The costs of the infrastructures are rather high because you work with expensive machines in a radioactive environment. Often when doing tracer research, you need the presence of a cyclotron nearby, so these are big equipments. And for example, with FDG, to be sure that you don't see artifacts related to inter-individual variabilities linked to the metabolism of each animal, you have to fast your animals. So this can induce additional toxicity for treatment studies. The example I will show using a PET is an example using a CXCR4 tracer. CXCR4 is a cell receptor that has been shown to be a target and a biomarker of metastasis. assisting diagnosis, staging, and therapeutic monitoring. It's overexpressed in several cancers. Its expression is correlated with the probability of metastasize, and it is also overexpressed in metastasis themselves. The stem cell mobilizing agent AMD 3100, called Plerixafor, is a compound that is currently under development against CXCR4. It has high affinity and has been labeled with copper-64, an isotope that can be used for PET imaging, in a study published by Nimagada et al. In Cancer Research in 2010. They have chosen 2 cancer models with the differential expression of CXCR4 measured by immunochemistry and FACS. They implanted tumor cells subcutaneously on both sides of the thorax of mice and perform a PET-CT imaging experiment with the copper-64 AMD 3100 to confirm its affinity to the target in vivo. In another experiment, they showed that the tracer was a good tool to monitor the progression of metastasis in the lung in a model using breast cancer cells injected intravenously. The signal increase in the lungs was about 2-fold compared with the control animals, and confirmed these results by immunohistochemistry. This signal difference in tumor versus control is not very high, mainly because CXCR4 is expressed in normal lung tissue, as you see here. This broad expression pattern is the cause for elevated toxicity in the development of anti-CXCR4 drugs, but would not represent a major drawback for an imaging tracer, as the doses that are injected are very low compared to therapeutic doses. First, just a summary of the different imaging modalities included what I've talked about and what I've not talked about, included computed tomography and ultrasound. They all have different properties in terms of sensitivity and in terms of resolution, ranging from MRI, which is mainly the least sensitive approach, to optical imaging. And the properties change. You also get information that range from anatomy to molecular information. And the properties change when using a contrast agent. It adds a little bit to the spectrum of applications. So you get different spatial resolution and temporal resolution in terms of what you want in a metastasis project. You get different invasiveness, you get different number of available tracers, and you also get different whole body imaging capabilities. So this is also the purpose of this slide. This is an example of the dissemination sites that are commonly observed. So different cancers are known to have preferential sites of metastasis. So I've tried to make a stand here on which modality would be better to image which localization. So, for example, with bioluminescence imaging, you get quite good whole body imaging properties, and you also good to image lungs and bones. For brain, it's a little bit more difficult because you have the skull, and the liver is mainly opaque to light, so you get signal from the outer surface of the liver, but not from the inner liver. With MRI, you typically get good results with brain. Bones is okay. In lungs, the problem is that you lack signal because in MRI you basically image water. But there are applications in which you can inhale a contrast agent in lungs, and it increases the usability of this technique in the lungs. In liver, it's a good modality to image liver. MRI is quite sensitive to movement, so you really have to be careful doing small animal imaging studies. because there is movement. You have to synchronize your acquisition with the breathing of the animal. It's a little bit difficult to do whole body imaging with MRI because the field of view is limited. So you typically need several images, several data sets to represent the whole body. With SPECT and PET, you can basically image anywhere in the body. You don't have much limitations, except maybe in the abdomen where the background signal can be quite high. So very often the signal you get from the abdomen is very difficult to interpret. You also get, depending on your machine, whole body capabilities for some machines. And CT and ultrasound are also good. CT is especially good for bones. For liver, brain is okay, but the contrast is not very good, and it gives very nice whole body images. Ultrasound, I haven't described it, but you can't get whole body images at all with ultrasound. So it's good for soft tissues like liver, and not so good for bones or brain because, again, of the skull. So as a conclusion, in this webinar, we have seen that there are ways to model and to evaluate the metastatic processes. One thing is for sure, metastatic disease remains fatal, and we need new compounds to progress towards better efficacy. We believe that progress can be made in a number of areas. New models need to be developed, in particular models that mimic the human immune system and the human tumoral microenvironment can be of help. For example, a project is ongoing at OncoDesign where we are trying to humanize the liver of mice by injecting human hepatocytes under optimized conditions. We also need better readouts to monitor the efficacy of novel treatments. We have presented some of the tools that might be available in the future, but many more are being developed. We believe that imaging has a great part in this, and that it has the potential to help us understand at the molecular level what is happening when treating. Because it's non-invasive, we can repeat measurements and study the time course of event. Because it's localized, we can understand the spatial heterogeneity of tumor response. Finally, we need to detect earlier the onset of the metastatic process. This can be done, for example, by being able to detect cancer stem cells or circulating tumor cells. This can also be done by monitoring non-malignant conditions that may be associated with tumors entering into the metastatic phase, like inflammation or hypoxia. Thank you very much. We are now ready to take any questions that you may have.
Xavier and Caroline, thank you both very much for that presentation. It's really interesting to me to see the different benefits and drawbacks of each imaging modality. Do you actually have access to all those imaging modalities?
Yes, we through a collaborative project that we started 4 to 5 years ago in Dijon, we have access to all the modalities that I mentioned, from nuclear imaging to MRI, CT, and ultrasound. So we've built a collaborative platform called FarmImage, in which the scientists in Dijon and in the area around collaborate to use these imaging technologies to understand the pharmacology of anti-cancer drugs and also in other therapeutic applications.
Okay. We will take questions from the audience. Please go ahead and type your questions in the Q&A panel. We have already received a few. Let's see, the first question, are there ways of using bioluminescence imaging for patient-derived xenografts?
Of course. Yeah, oh no. No, I'm sorry. Yeah, we would need to be able to transfect well, this is typically used for cell lines, and this we need a transfection. But the optical imaging in general can be useful. Maybe not bioluminescence, but fluorescent imaging has no such need. We don't need a transfected model expressing luciferase. We can just inject a fluorescent tracer and use it. It can be targeted towards a specific target, like, for example, there are tracers towards alpha V beta 3 integrins, and they home to their target, and we can see them in the body. One of the commonly known limitations of fluorescence technique is that you get quite a high background signal from the animal, and the tissue penetration is also a bit less good than it can be with bioluminescence imaging. So my answer shortly is no, but don't give up on the optical imaging, because there are also other techniques.
Okay. You showed some metastatic models with the PC3 prostate cancer cell line. Do you have any other metastatic or I guess just regular prostate cancer models with other cell lines?
For prostate cancer model, with spreading in metastatic dissemination, no, it's only for PC3 human prostate tumor cell line with bone metastasis by IV injection.
Maybe this can be a question that can be sent to us by email later, and we can answer directly.
But for prostate, I see only PC3 cancer model.
Okay, very good, thank you. Let's see here. With small metastases, is there enough sensitivity with these imaging techniques to pick up these sort of micrometastases? What is the limit, the size limit to pick up these metastases? Is that something that's known in terms of tumor volume or cell number?
Yeah. In my next to last slide, I described the modalities in terms of sensitivity. And for these applications where you need extra sensitivity, I would go for bioluminescence imaging, for which you can detect up to hundreds of cells. And this, I guess, would be the most sensitive approach.
Okay.
Depending on the depth, of course, because to be sensitive, you need to be quite close to the body, to the outer surface of the body.
Okay. We have a question here about study design. So how many animals are typically included in an imaging study, and does that differ by modality?
Yes, it does. It depends on mainly on the time you need to do one imaging on one animal. So as I try to describe, it can take from several minutes or seconds even with bioluminescence imaging, up to 1 hour or so with nuclear imaging or some MRI protocols. What we need to do at OncoDesign is to limit this time, and we consider that 1 hour is the longest time we can keep a mouse anesthetized and keep it in good shape and in good condition to be able to repeat the measure and so on. So depending on the time it takes, we can image from 6 to 8 animals per day up to 150.
Okay, very good. Thank you. We are out of time for questions. We do have a couple that weren't answered, and we will follow up with those by email. Caroline and Xavier, thank you both very much for your great talk, and thank you everyone for attending.
Thank you.
Goodbye.
Thank you. Bye.
Thank you. Bye.